Just give a brief intro to the talk. We might as well get started. So really delighted to have you all here today from all over the world. I think that we're all here because we're passionate about longevity, we're passionate about dogs, and I think we all can agree that the kind of multi-millennia history between dogs and humans is really taking a fascinating turn in frontline longevity research, which is where Matt is coming to play. So we have Matt Kaeberlein here today. He's going to be giving a talk. Before I introduce Matt, I just want to give my quick spiel. Welcome to the Oxford Society of Aging and Longevity. We're Oxford University's Student Center for Longevity Research, Education, and Advocacy. We host lots of wonderful talks on the topic of longevity. We're very privileged to always have incredible speakers talking about different aspects of healthy aging, longevity, frontline science, and also the social, political, economic ramifications about this radical shift in healthcare. So if you're interested in longevity, healthy aging, healthcare, all of that, make sure, if you're not already subscribed, to subscribe to our newsletter, join our talks. If at any point you are interested to learn more about the Society or get involved, reach out to me or any of the other committee members. We're always here for you guys. So without much ado, delighted to have Matt Kaeberlein join us today. Matt is an incredible force in the longevity space and wears lots of different hats. I'm going to read out his bio, which is quite the handful.
You don't need to do that.
Well, I mean, you're probably sick of hearing it, but there's probably going to be a lot of people that aren't familiar with the different facets of your work, which dog aging is just one part. So Dr. Matt Kaeberlein is the Chief Science Officer at Optispan Ventures and an affiliate professor of oral health sciences at the University of Washington. Dr. Kaeberlein's research interests are focused on understanding biological mechanisms of aging in order to facilitate translational interventions that promote health span and improve quality of life for people and companion animals. He's fellow of the American Association for the Advancement of Science, the American Aging Association, and the Gerontological Society of America. Dr. Kaeberlein has published more than 250 papers in the field of aging biology and has held several prominent leadership positions, including founder director of the University of Washington Healthy Aging and Longevity Research Institute, director of the NIH Nathan Schock Center of Excellence in the Basic Biology of Aging, director of the Biological Mechanisms of Healthy Aging Training Program, founder and co-director of the Dog Aging Project, and CEO and chair of the American Aging Association. So as you guys can all see, Matt is involved in a host of different activities. Today's talk is really going to be focused around the Dog Aging Project. However, at the end, we're going to have a Q&A where you can ask more questions about the Dog Aging Project, share your feedback, and also ask questions about some of the other research and initiatives that Matt's involved in. So if you've got some questions, please drop them in the chat during Matt's talk. We'll have a Q&A at the end. And then at the end, we'll kind of come back to them and have a bit of a discussion. So without further ado, I'm going to pass it over to Matt. Matt, I believe you've got some slides, so I'm going to stop sharing my screen.
Okay, great. Thank you for that kind introduction. It says host disabled participant screen sharing. So I don't know.
Let me make it
means you don't want me to share my slides. I can do a chalk talk, Zoom chalk
we
talk.
could here. I just need to make you a host. You are now the master of this call.
All right. That's great.
And
Let's see if I can pull up the right slides. All right, this looks right. Dog Aging Project. Okay, great. So thank you, first of all, for that kind introduction. I'm seeing people who want to be admitted. Do I need to admit them now, or can you still do that? I admitted one person, but just want to know if you want me to be the one to let people in. It's okay. You're muted. I'll figure it out. We'll make it
Oh,
work.
sorry. That's a yes, Matt, if you don't mind. I'm very sorry about that.
Okay. I can do more than one thing, hopefully. If I can figure out how to close this window. All right. So anyways, thank you for the introduction and for the invitation to give this presentation. If I stop talking and stare in a confused way at my screen for a minute, it's because I'm admitting people, but we'll muddle through this. So I'm going to talk about the dog aging project today. My hope is it'll take about half an hour, and then that should leave 20 minutes or so for discussion. And I'm happy to talk about anything that you all want to talk about, preferably related to the biology of aging in some way, shape, or form, but really open to any discussion topics in the field that people are interested in talking about. So let's see now. Okay, so the dog aging project, I guess it's about 10 years old now or so, I would say, at least my role in the dog aging project. And really for me, in large part, this project stemmed from a question that I think many of us in the field have been asking ourselves for a while, which is how do we start to bring some of what we have learned in the laboratory out into the real world? And I think there are lots of smart people thinking about this and taking different approaches. The approach that I have settled on, one of the approaches, I guess I should say, that I have settled on, is the idea that I think our companion animals and companion dogs in particular provide a really unique and powerful path to translational geroscience, or to bring some of what we've learned about the biology of aging into the real world, to understand aging better in the real world. The dog in this picture here is actually my dog, Dobby, who I love very much. And I will show a couple more pictures of him because, one of the great things about working on the dog aging project and giving talks about it is I get to show pictures of my dog when I give these presentations. So the idea here, though, you'll note that there's a double-headed arrow between Dobby and the human in this case. And I put that there intentionally because I think for me, what we learn in the dog aging project is certainly not only about what we can apply to humans. I want to make the point explicitly that I believe that understanding and hopefully being able to intervene in the biology of aging in companion animals to extend health span and potentially lifespan has intrinsic value in and of itself, kind of independent in some ways even of how that moves us forward in terms of being able to do the same thing for human health. And so for me, a big part of this is because I've been studying the biology of aging for 20 plus years now, but I've also been a dog person my entire life. And I recognize how important our companion animals are to many of us. I think many of us consider our companion animals to be part of our families. And the opportunity to have a positive impact on health and well-being in companion animals, I believe, has significant intrinsic value. So there are really two kind of overarching goals of the dog aging project. And I'm going to talk about both of them because they're sort of independent pieces of the dog aging project. The first one is to understand the biology of aging. And we do that through a large-scale longitudinal study of aging. Sorry, I'm getting distracted here with the waiting room and not being able to get rid of these windows. Wow, now I'm drawing stuff. This is crazy. All right, sorry, everyone. This is a glitch in the system. So one goal is to understand the biology of aging. I'm going to talk about the longitudinal study of aging that we are carrying out to accomplish that. And the other goal is to do something about aging. And I'll talk about a clinical trial that we have ongoing to hopefully have an impact on longevity, health, and well-being in companion animals. So I want to start, though, by taking a step back and addressing the question of why even think about the biology of aging in dogs in the first place. I think there are lots of reasons why companion animals, pet dogs, make sense in this context. We all know that dogs age very much like people do. For any of you who have had dogs, you will recognize that dogs develop most of the same age-related functional declines and diseases that people do. And importantly, those are age-related in dogs. But dogs age much more rapidly than people do. And we're all kind of familiar with this idea of, one human year is about 7 dog years. Another way to say that is just dogs age about 7 times faster than people do biologically. And so that more rapid aging process means that we have the opportunity to actually study, understand, maybe even intervene in the aging process in dogs in a time frame that's amenable to scientific research. Dogs also have this very unique sort of genetic and phenotypic diversity. So dogs are made up of a whole bunch of pure breed dogs, which you can kind of think of like inbred mouse strains, where they're very genetically homogeneous. But then on top of that, there's this complicated, genetically heterogeneous mixed breed population, which is very sort of unique genetic architecture among animals. Dogs are also quite phenotypically diverse. So if you think about almost any phenotype of relevance, dogs are at least as diverse, if not more diverse than humans are. And body size is a really easy one to think of. And just consider the difference between a Great Dane and a Chihuahua. So that combination of genetic and phenotypic diversity is quite powerful for mapping traits onto genotype. One of the ones that I think is underappreciated, but particularly relevant for the field of aging research is that dogs share the human environment, which is just simply something we cannot capture in laboratory animals. In fact, we do almost everything we can to control the environment in the laboratory. But of course, humans live in this very complicated, heterogeneous environment, and we all have our own unique environment. And certainly that plays an important role in health outcomes during aging and in the biology of aging. And the graphic here is meant to illustrate that. And again, I'll apologize because I can't actually see the graphics because I have this participant window open that I can't get rid of. So I'll do my best to put the pointer in the right place. But just like in humans, there are rich dogs, and this you may not be able to tell, but this is actually the doghouse. This is the dog. There are economically challenged dogs. There are dogs with what we might consider healthy lifestyle habits, and there are dogs that have potentially unhealthy lifestyle habits. And of course, dogs don't actually smoke, but dogs do breathe the same air that we breathe. They drink tap water typically. And so we can capture that environmental diversity and some of the factors that may play a role in the biological aging process in the environment. I've already mentioned how important dogs are to many, many people. And I think this project kind of uniquely can engage people in citizen science or community science. And I've also already alluded to the fact that it's kind of independent of what we learn about aging in humans. Being able to improve the quality of life for companion animals has its own intrinsic value in the quality of life for the people and the pets. Okay, so I thought I'd start by just giving you an overall idea of the current structure of the dog aging project and how the project is set up. So the largest bucket in the dog aging project is the longitudinal study of aging. And all I mean by longitudinal study of aging is we are following dogs over time throughout their lives, collecting data from the owners and the dogs at multiple time points over their lives. And so to be part of the dog aging project longitudinal study of aging, owners really only have to do one thing, and that's go to the website, nominate their dog to participate, and complete the comprehensive, what we call health and life experiences survey. And that's what the HLES here refers to. This is a pretty extensive survey instrument that tries to capture as much information about the dog's health history, current environment as we possibly can. So it takes people, anywhere from an hour and a half to 2 hours to get all the way through this survey instrument. Once owners have completed HLES, they are then become part of the dog aging project pack, and they are asked annually to update that survey instrument. We have now, I think, slightly over 45,000 dogs, all in the United States for now. who are part of the Dog Aging Project pack. So it is a very, very large data set. After completing HLES, owners are also asked to complete another survey, which I'll come back and talk more about in a little bit, called the Canine Social and Learned Behavior Survey. That's CSLB. For now, just think of it as a cognitive assessment, and I'll tell you more about that in a few minutes. Owners are also asked to provide electronic veterinary medical records. If they are able to provide the veterinary electronic medical records, they then become eligible for these other groups within the pack. And those are what we call the sampled cohorts or sampled groups. And there are several of those. Oh man, did I just stop sharing, or can you guys still see it? Somebody give me
stop sharing.
Sorry, everyone. This is a little clunky because I'm trying to deal with multiple windows here and admit people. Okay, does that look good?
Perfect. Thank you.
Yes.
Yeah.
Okay. All right. Cool. So, among the sampled groups, the foundation group is the largest, and that's the group that gets their whole genome sequence. And there are 10,000 dogs currently allocated to the foundation group. So 10,000 canine genomes sequenced, and then within that group, about 1,000 dogs are selected for the precision group. This you can think of as the systems biology group. So every year, the owners are sent a sample kit that they take to their veterinarian for their annual visit to collect annual samples for microbiome, epigenome, metabolome, things like that. So every year we get that sort of high dimensional comprehensive data. Whoops. So that's the precision group. Again, I apologize for the delay in my talking, trying to deal with these windows. All right, so then we have a specific study dedicated to cognitive function, which I'll tell you more about, the brain health study. Another study really dedicated to trying to understand the genetics of extreme longevity. These dogs, that's the centenarian group here. These dogs obviously don't live to be 100 years old, but these are dogs that live exceptionally long for their body size. And we want to understand, are there genetic factors that we can correlate with those very positive longevity outcomes? And then the last group is the clinical trial group, the test of rapamycin in aging dogs, or triad, which again, I'll tell you a little bit more about that in a few minutes. Okay, so this is just to show you a little bit more about the kind of information we get on the dogs in the pack and the geographic distribution. The geographic distribution is more or less what you would expect for dogs in the United States, overrepresented for urban areas, not surprisingly, overrepresented for the coasts, but I think overall have a pretty good geographic distribution of dogs around the United States. This is Sage. Beautiful dog, 25,000th member of the pack. I keep saying once we get up to 50,000, I'll change the picture and put our 50,000th member up there. And you can see the kinds of data that we collect for all the dogs in the pack. Sorry, the numbers you can tell are a little bit off because they keep going up. So again, we have about 45,000 dogs in the pack right now. And I just show this to make the point that we are still really at the tip of the iceberg in analyzing this data. So we have done, I think, a pretty comprehensive analysis to the first year's data, which was really cross-sectional, meaning only one time point. And I'll show you a little bit of some of the insights that we've gotten from that first year's data. Now the project is a true longitudinal project, meaning we have collected data on many, many thousands of dogs for 2 years, some for 3 years. And so we can start to see how things are changing with time in these dogs as they're getting older. But there's so much data that there's just a huge amount of opportunity here, I think, to do really deep dives in really interesting ways. And so that brings me to a point that I just want to share with everybody, which one of our core values at the Dog Aging Project is that we are an open science, community science project. So we published a paper in Nature last year, kind of describing the structure of the project, the kinds of data that are available, and how the scientific community can get access to that data. Every year, we do a data release for all the data that we've collected up to that point. Very simple data use agreement on the website. So if any of you are data scientists or you just love dogs and want to dive into the data, you can get access to the data. Go to the website, dogagingproject.org. complete that with what should be a very simple data use agreement, and you can get access to the data. And I say that because, again, we recognize there's just so many different things that can be done with this very rich data set that our team simply isn't going to have time to do. So definitely would encourage you to take advantage of that if that's something that's of interest to you. So I want to just give you kind of an overview of some of the data from the pack. And this is from the first year's data release, which was about 25,000 plus dogs. So you can see what our age distribution looks like. Again, this is pretty good. We think this is, relatively representative of the companion dog population in the United States. We know we're underrepresented for very young dogs, and we're working to enroll more puppies. But again, we have pretty good age distribution, pretty good size distribution of dogs, lots of dogs in the different size categories. Roughly half male and half female. However, it is worth noting that there are really four sexes in dogs in a sense. There are intact males and females and sterilized males and females. And, much again, like the companion animal population in the United States, we are dramatically overrepresented for sterilized dogs compared to intact dogs. And, that's just going to be in some ways a nature of the companion animal population. However, I think it is worth recognizing that it is very likely, if not certain, that sterilization is going to affect some aspects of biological aging. And so again, we are working to increase the number of dogs we have that are intact within our overall cohort. And again, pretty well represented between purebred and mixed breed dogs. So this is showing you owner-reported health status of the dogs. And I'm gonna try to say that word a couple of times as I'm talking about the data. It is important to keep in mind that the data from the Health and Life Experiences Survey is owner-reported. And there are strengths and weaknesses of owner-reported data. One of the weaknesses is that owners, it's not laboratory measured, right? Owners may or may not accurately report information about their dog. So this is the owner-reported health status of the dogs. And you can see most of the dogs in the Dog Aging Project pack, the owners feel are in pretty good health. And we think this is pretty accurate. I should say, overall, we're pretty happy with the quality of the owner-reported data. But the owners are pretty confident that their dogs are in good health. And we believe that we probably have, to some extent, a healthy cohort bias. That's going to be natural. It's less likely that owners are going to sign their dogs up for a long-term longitudinal study of aging if their dog is already sick. But we expect that dogs that are in the pack over the coming years will develop age-related diseases and functional declines and become, less healthy. Having said that, even still, we have this sort of general owner-reported health status, but we also have information about prior disease diagnoses within the pack. And despite the fact that the vast majority of owners think their dogs are in excellent health, there are a lot of disease diagnoses for dogs in the pack. And so this again comes from the Health and Life Experiences Survey. So this is owner-reported disease diagnoses. This is not data yet extracted from the electronic medical records, but you can see there are a lot of disease diagnoses among dogs in the pack. And I'm not going to take you through the data, but for each of these, we have additional information. So for example, cancer or tumors, if an owner says, my dog has been previously diagnosed with cancer, then they get taken to a branching survey tree that asks them more about the cancer. What tissue was it in? What, how advanced was it at the time of diagnosis? What was the treatment modality? So we have that additional information also from the Health and Life Experiences Survey. But this gives you a feel for, again, lots and lots of disease diagnoses already in the pack that even from a cross-sectional perspective, you can start to do some correlative analyses on. So then one thing we can do then is a little bit of a sanity check and do some very simple correlations, one of which was to ask, okay, what happens to owner-reported walking speed as a function of the age of their dog? And as you might expect, walking speed goes down with age in dogs, just like in people. This is not earth-shattering by any stretch of the imagination, but it is, like I said, a nice sanity check. If we saw that the dogs were getting faster with age, we'd be worried, but this makes sense, and it's nice to see that this is matching what we would expect. We can also ask some questions about how does the frequency of prior disease diagnoses change as a function of age. And so in this case, we're looking at all forms of cancer. This is broken out by body size. And so what's shown, and I'll just note, this is a semi-log plot. So this is the log of incidence versus linear age. And as you would expect, the risk of a prior diagnosis of cancer in dogs goes up essentially exponentially with age. And that's exactly what we see in humans. So this makes sense to us. And again, in some ways, this is a bit of a sanity check, but it's really nice to see that the data is matching what we would expect for a disease of aging, in the sense that it goes up exponentially with age. And what's interesting is this is broken out by size, but if you compare all of the size categories, what you see is that at every age, big dogs are at a higher risk of having been diagnosed than small dogs are. And again, this makes sense. We believe that in most ways, big dogs are aging biologically faster than small dogs are. And certainly for life expectancy, big dogs have a shorter life expectancy than small dogs do. Now, I will come back and show you one specific case that doesn't match this trend, but for most age-related functional declines and diseases, big dogs appear to age faster than small dogs do. Okay, so what are some interesting questions you can ask with a data set like this? And there are lots of them that the team has set out to try to look at. One that I was particularly interested in was this question around caloric restriction, amount of food, fasting, time of feeding, and would there be any correlations with health outcomes in pet dogs? And so there were a variety of ways that we could have approached this. The way that we decided to look at the data first was to ask a very simple question, which is, does the number of times per day that a dog is fed correlate in any way with age-related disease diagnoses, prior disease diagnoses, okay? And the reason for looking at that is because, there's a lot of interest in the field around intermittent fasting and time-restricted feeding. And when you actually look at the mouse studies, the data are despite what will commonly be, put out there, the data are really not very convincing that intermittent fasting or time-restricted feeding have any effect on longevity once you control for caloric consumption. So certainly if the mice are calorically restricted, you can find evidence that lifespan is extended and health is improved. Once you control for caloric consumption, though, those effects pretty much essentially go away in mice. So we thought, well, this might be an interesting opportunity to look in a natural population, simply because some owners feed their dog once a day, some owners feed their dogs twice a day, some owners feed their dogs 3 times a day. Believe it or not, a significant fraction of owners actually give their dog free access to food, which is shown as ad libitum here. So what's shown in the data, the plot here, is just within the pack, how many dogs fall into each of those categories. Now, I should say, I think the ad libitum group is kind of self-selecting. There are many dogs where if you gave them free access to unlimited food, they would blow up into a blimp. But certainly among the once, twice, and three times a day categories, we've got enough dogs in each group that we could ask very simple questions like, is there any difference in prior disease diagnoses for dogs fed once a day compared to dogs fed more than once a day? And so that was the first order question that we asked, was you take all the dogs in the once a day group and you compare that to dogs in the more than once a day group, is there any difference in prior disease diagnoses? And I didn't expect this to work based on my interpretation of the mouse data that I just told you about. I was wrong. So we looked at 10 different categories, 9 of which are shown here. Cognitive function was the 10th. And so for 7 out of the 10, 6 of which are shown here, there was a statistically significant lower probability of a prior diagnosis within that disease category in the dogs fed once a day compared to the dogs fed more than once a day. And in some cases, the effect sizes were pretty big, like for liver and pancreas disease. So again, correlation does not equal causation, but this looks like a pretty interesting result, right? That in every case, the trend was going in the right direction. And in 6 of the 9 shown here, and also for cognitive function, it was statistically significant. The dogs fed once a day were less likely to have been previously diagnosed with that particular disease outcome. So there are other explanations. This doesn't have to mean that once a day feeding causes better health. Could be that dogs fed once a day are less likely to be obese and therefore less likely to develop age-related disease. But it does certainly look like there is some relationship here that is worth understanding in more detail. And so there are people on the team working on trying to understand that more at this point. But as I said, correlation is not causation, and trying to get the media to appreciate that is always a challenge. So as you can imagine, there was lots of interest in this story, and sometimes over interpretation, I would say. Okay, so another thing that we are very interested in, as I sort of have already alluded to, is cognitive function within the Dog Aging Project. We have a dedicated brain health study funded by a separate grant from the National Institute on Aging. Some of you may know this already, but some dogs do develop dementia. It is age-related in dogs. It's a disorder called canine cognitive dysfunction. And dogs with canine cognitive dysfunction do show some of the same molecular and neuropathological hallmarks as humans with Alzheimer's disease. Although I think the consensus is that it probably is not a true Alzheimer's disease dementia, but they certainly show some of the same hallmarks. So we are measuring, as I've already mentioned, cognitive function annually, and I'll show you how we do that in a minute through this canine social and learned behavior survey. We are also developing games that owners can play with their dogs that are meant to assess different domains of cognitive function. And we're very interested in the genetic and environmental factors that may be predictors of dementia in dogs, also interested in blood biomarkers, as well as further assessing to what extent do dogs with dementia show proteomic signatures, neuropathological signatures that may be similar to people who develop dementia, and does it look like Alzheimer's disease? Does it look like other age-related dementias in people? So that's all part of this overall brain health study within the Dog Aging Project. I want to talk just briefly about this canine social and learned behavior tool. So this is a validated survey instrument, very short, only takes owners about 10 minutes to complete. And it's been shown that it is highly correlated with a veterinarian diagnosis of canine cognitive dysfunction. And the way this survey works is you complete the questions and then you sum up the score. And if the score is above 40, that is clinically diagnostic for canine cognitive dysfunction, about 95% specificity. So the tool, though, has been used primarily as this binary tool, a yes/no. Does the dog have dementia or does it not have dementia? We were very interested in the question of, could this be used as a continuous tool to maybe predict dogs that had mild cognitive impairment or early symptoms of dementia? And so we, as I mentioned, this is part of the annual survey that all the owners in the Dog Aging Project take. And so these are showing you the CSLB scores for all of those first 25,000-ish dogs in the Dog Aging Project. And what I want you to appreciate is the vast majority of dogs are below the diagnostic threshold. Sorry, I'm going to apologize. I think I said 40, 50 is the diagnostic threshold. 40, we think, might be sort of a sign of mild cognitive impairment, but that's very speculative. We have a couple hundred dogs that are above 50. We have many dogs that are between 40 and 50. But again, the vast majority of dogs in the overall pack fall in the well below the diagnostic threshold for dementia. So you can ask the question now, what does this look like as a function of age? Okay, so this is all the dogs in the pack. If we break it out by age, if this is a continuous tool, you would expect that we should see an age-related increase in score. In other words, a decline in cognitive function with age. So we looked at that, and indeed, that's exactly what you see. And indeed, it looks like an exponential change with age across body sizes. So in both male and female dogs, as a function of age, the CSLB score goes up below the diagnostic threshold, at least for the average, but it goes up what appears to be essentially exponentially with age. And so this tells us that this may very well be reflecting in a continuous way changes in cognitive function with age. The interesting thing here is that there really is no size effect. So if you think back to that cancer data that I showed you, where at every age, big dogs were at a higher risk of developing or having been diagnosed with cancer, here there doesn't really seem to be a size effect. In other words, big dogs aren't really at any higher risk of developing dementia, and that's true, than small dogs are. So maybe this is telling us something interesting about brain aging. Again, speculative. I don't know the mechanism there, but it's interesting to think about, and this is something we want to study further. Okay, so we've also looked at what else is correlated with cognitive decline or dementia in dogs. Age is the greatest risk factor for dementia in dogs. Again, that makes sense. That's what we would expect. But once we get past age, it's interesting. Exercise or activity is the second greatest correlate with risk of dementia in dogs. Sterilization seems to be correlated with risk of dementia in dogs. And then prior history of some other neurological eye and ear disorders is correlated with greater risk of dementia in dogs. And again, all of these things are really interesting. I don't know about data for sterilization in people, but certainly exercise, this matches what we think is the case in people. And there is some evidence in people that hearing loss, vision loss may be an early correlated, at least, with dementia in people. So some interesting potential parallels there. And the New York Times picked this up, and it was cool. My Dobby got to be in the New York Times, actually his second time in being featured in the New York Times, which was pretty awesome. So again, clearly a lot of interest in this topic and understanding dementia in dogs and its relationship to dementia in people. Okay, so now I'm going to switch gears and just spend the last 10 minutes or so talking about the clinical trial, which is the test of rapamycin in aging dogs. And again, the longitudinal study is really designed to understand what are the most important genetic and environmental factors that influence aging and health outcomes in dogs. The clinical trial is really to test, can we actually have an impact on biological aging in dogs? And the clinical trial that we have going now is designed to test specifically whether the drug rapamycin can impact aging in dogs. And the reason for testing rapamycin is that it is the most effective longevity drug that we know about from preclinical studies in laboratory animals. And because dogs age rapidly, we have the opportunity to answer the question in a few years as opposed to in a few decades, which is really what it would take if you really wanted to look at lifespan and age-related health outcomes in general in people. So again, test of rapamycin in aging dog. Why rapamycin? Rapamycin has an interesting backstory. I'm not going to take you through it, but for those of you who are interested, you can look it up on Google. It was discovered on Easter Island. That's where the drug gets its name from, Rapa Nui. Importantly, it's been FDA approved now for more than 20 years. So we know a lot about clinical use of rapamycin in people. We know a little bit about clinical use of rapamycin in dogs. And that clinical information is very valuable when you think about trying to design this kind of a clinical trial, because it was able to guide us on dosing and things like that. I already mentioned it's the most effective longevity drug. In mice, it increases lifespan up to about 30%. And again, from the perspective of a translational clinical trial, it's really important that in mice, at least, you can start the treatment in middle age, about the mouse equivalent of a 60-year-old person, and still get benefits. And so that gives us some confidence that we may be able to have a similar effect in dogs and eventually in people. And also importantly in mice, it appears to broadly delay biological aging, functional declines, molecular declines in many, many tissues and organs, a subset of which are shown here at the right. And in at least 4 places, you can actually reverse age-related functional declines in mice. And those are shown in red. And again, from a clinical trial perspective, it's much more it's easier to imagine measuring a change where you can actually see an improvement potentially in an age-related functional decline, as opposed to trying to measure a change in the slope of the decline. So again, we don't know yet in dogs or people to what extent will this translate, but it gives us some level of confidence that there's an opportunity here. So I just wanted to show this one piece of data from mice. This is actually work from my lab several years ago that was done by Alessandro Bito, a former postdoc in the lab, now a faculty member at the University of Washington, where we did a very simple experiment. We took mice at 20 months of age. We gave them rapamycin for 3 months. Then we stopped the treatment. Then we put them back on the shelf and just asked what's the effect on lifespan compared to mice that got the vehicle. And in this particular group, we saw an increase in life expectancy of about 60%. And so again, we don't know whether that's going to translate through to dogs, but if it does, that's about 2 to 3 years for a typical middle-sized dog. So it's the data like this that we were able to base our power calculations on for trying to figure out how many dogs do we need in the study, how long should the dogs be treated with rapamycin, and what sort of effect size might we be able to expect. Okay, so this is the design of Triad, test of rapamycin in aging dogs. The study is designed to enroll 580 larger middle-aged dogs. They have to be at least 40 pounds and at least 7 years old at the time of randomization. They can't be sick. This is a really important feature of the study. The dogs have to be of normal health status for their age, because we really want this to be a study of normal aging, not a study of disease. Half the dogs will get placebo, half the dogs will get rapamycin. The dose is 0.15 milligrams per kilogram given orally by the owners once each week. So 1 year treatment period, 2 years of follow-up, so 3 years in the study for each dog. And lifespan is the primary endpoint. So our statistical power calculations were designed to detect a 9% change in lifespan. That's the primary endpoint. But of course, we're interested in health span metrics, as many as we can measure. And so we have as secondary endpoints a variety of health span metrics that we are looking at over the course of the study. We've completed 2 small clinical trials. The first one was for 10 weeks. The second one was for 6 months. Key takeaways from those trials were no significant side effects detected, increased owner-reported activity in both studies. And in the first study, we found some evidence for age-related improvements in heart function that paralleled what had previously been reported in mice. This is just a schematic giving you sort of an overview of the trial design. Again, I mentioned the dogs come in for a pre-screening visit. They have to be healthy at the time of randomization. If they pass that pre-screen, they are then enrolled and randomized, block randomized at each clinical site, half placebo, half rapamycin. Within the rapamycin arm, well, within both arms, I guess, dogs will either get echocardiogram, so cardiac analyses every visit, or neurological analyses at every visit. So we have those sort of 2 separate health span metrics as secondary endpoints. Dogs come back every 6 months to the primary veterinary clinic. At every visit, we collect either heart data or neurological data, and then we also collect metabolome, microbiome, epigenome samples, blood chemistry, full veterinary exam, really trying to get as much data on these dogs as we can. Owner surveys are completed regularly throughout the entire study period. After 12 months, the dogs come off of treatment, but they remain blinded for the 2-year follow-up period. And then at the end of the 3 years, the dogs come out of the study. So once every dog has completed the 3 years, the study will be completed, we will unblind, and hopefully we will be able to answer the question of, does rapamycin slow aging in dogs? This is showing you our current clinical sites. This is actually a little bit out of date. We have added additional clinical sites, and we're up to, I think, 18 or 19 clinical sites at the moment. So, so we're actively enrolling and randomizing dogs in the study right now. And I'll just finish by mentioning that I'm very fortunate to get to talk about this, but really this couldn't happen without the awesome team that is doing all of the work. I don't have time to mention everybody in the dog aging project. It's become a very large project, many different institutions involved. I do want to specifically mention Daniel Promislow, who along with me is the co-director of the project. Daniel is the primary investigator on the NIA-funded U19 that funds most of the longitudinal study of aging and much of the triad clinical trial. I also want to mention Kate Creevy at Texas A&M University, who is our chief veterinary officer. And it's really Daniel, Katie, and I that are the founders of the Dog Aging Project and that represent the leadership of the team. But of course, it is this much larger group of people that that really make the team what it is and really deserve the credit for what we've been able to accomplish to date. So with that, I will stop talking and be happy to answer any questions that people might have. Thank you.
Thank you so much, Matt. That was really, truly fascinating and excellent. I've got tons of questions, but I'd like to hold them off because we've had some really excellent questions come up in the chat. I'm just going to call on a couple of people who've asked some questions in the chat. I'm going to start with Krister Kauppi. Do you want to ask your question?
Yes, I can ask my question here. I'm wondering how the dog aging project is financed.
Sure. So the Dog Aging Project is a mix of federal, United States government federal support through the National Institute on Aging and philanthropic support. So the Dog Aging Project has been fortunate to get two fairly large grants from the National Institute on Aging. One is the U19 that I mentioned that funds most of the longitudinal study, and then what's called an R34. It doesn't really matter what the letters and numbers are, but that's specifically to fund the brain health study component of the Dog Aging Project. And then we've also been very fortunate to get philanthropic support from some large donors who supported an expansion of the Rapamycin clinical trial to let us increase the power of that trial. And then probably, certainly hundreds, probably more than 1,000 small donors, who have each contributed, anywhere from $5 to $500 in support of the project. So very, very grateful, obviously, to all of the different sources of funding. To date, the Dog Aging Project has received no for-profit funding, if people are wondering. So it's all been, either federal grants or donor support.
And Matt, in terms of the participants in the study, in order to do these lab tests and things like that, are those tests covered by the Dog Aging Project? Okay.
Yeah, the owners are not asked to pay for anything. I will say, for the clinical trial in particular, where the owners bring their dogs to the veterinary clinics. So the clinical trial is sort of unique. For the other aspects of the Dog Aging Project, owners can go to their own veterinarian. We're not asking them really to do anything different. For the clinical trial, owners do have to commit to bringing their dogs to one of the partner clinical sites once every 6 months. We offer them a gift certificate, a gift card, sort of as compensation for the cost of their time and gas and things like that. But all of the veterinary care, all of that is paid for by the study.
Excellent. Yeah, because I was wondering maybe if it wasn't, it might bias towards certain type of owners that are more likely to take care of their dogs in a different way.
Yeah.
Wonderful. So we've got a question from Ajantha. Ajantha, would you like to ask your question to Matt?
Yeah, thanks. Thanks so much for that, Matt. That was really fascinating. I was wondering how, as you're taking these dogs from the DAP sort of cohort and taking them into other cohorts, how you're selecting them for all those other different cohorts, what you're looking at in particular, and I guess especially how you deal with all the different comorbidities that might come up as dogs get older as well.
Sure. Yeah. Great question. So I will be fairly general in my answer, in part because I'm not on the team so we had a lot of experts go through and figure out how we try to stratify these cohorts in a way where we get broad representation. So the real answer is I can't give you the details, but smart people did that work. So what I can tell you is first of all, it depends on the cohort. So for example, the centenarian cohort, the dogs that live longer than you would expect, right? You have to look at body size and prior demographic data to be able to come up with a statistical cutoff of how old is old enough to be statistically very unlikely to reach that age, right? So that was sort of how that was done. For the genome sequencing group, the foundation cohort, and the systems biology group, the precision cohort, there it was much more about trying to get appropriate diversity of genetics. So not being overrepresented for any particular purebred breeds, but also trying to pick enough dogs of enough purebred breeds that you had genetic representation across what we think is the diversity of genotypes in dogs. And then also making sure that you have, I think it turns out to be about 50% mixed breed dogs within those groups. Also trying to balance geographic distribution, socioeconomic, owner socioeconomic status. That gets back a little bit to, I think one of the points that was being made earlier about maybe there's biases within the owner groups for which dogs are in the dog aging project. We can only control that so much. There is in fact a bias there and we can't really control it, but we do want to take that into consideration when we're selecting dogs. So I think it was a mixture of all of those things and trying to come up with some sort of algorithm or bucket sizes for each of those buckets that we then try to fill. So we have numbers that we're trying to hit for the various buckets, and then we select dogs into those buckets, and once the bucket is full, we don't select any more dogs into that particular genetic age group bucket. For the clinical trial, it's a little bit different again in that we have very set criteria for the entrance criteria for the clinical trial. The dogs have to be at least 7 years old and between 40 and 110 pounds. And so, those criteria, any dogs that don't fall into that group are excluded from the outset. And then dogs that fall into that group, we then ask the owners, are you interested in participating in the clinical trial? So far, we're not, we have some criteria to try not to become too overrepresented for things like golden retrievers or labs, just because they're so common. But we're more interested in being able to fill out the trial with the enrollment criteria and less worried about trying to get, equal numbers of all the possible breeds and things like that. Because with 580 dogs, we're not going to be statistically powered to look at the effects of rapamycin across genetic backgrounds, for example. We know we're not going to have that statistical power. So we're not so concerned about that. Hopefully that gives you an idea, at least, of how we've approached it.
That was great. Thank you so much.
Great. Well, next I'd like to turn to Girish. You've asked a couple of great questions. I think some of them have been partially answered by Matt. But maybe do you want to bring up your question on biological age diagnostics? And in case Girish is offline, I'm just going to go ahead and read it out. Matt, what are your thoughts on the validity of using biological age diagnostics, epigenetic clocks, glycan age, eye age, etc., as endpoints in clinical trials? What will it take to validate them as robust endpoints moving forward? And we actually have another related question, which I might as well ask as part of this. Can we, and this is by Oleg Mashnov, can we create a way to measure aging that works for both people and dogs? Specifically, could we develop a transcriptomic aging clock using human tissue that, when applied to ortholog genes in dogs, correlates well with the age of those dogs? This could help us track dog aging that then might be translatable onto humans as they share the same measure of aging.
Yeah, well, those are both really good questions, and I will try not to get too far in the weeds because we could spend an hour talking about those questions because they're really, really complicated, really interesting. So what I would say around the biological aging diagnostics that were brought up, epigenetic age, glycan age, pheno age, right? There's a bunch of these things that are already out there. They each measure a part of biological aging. None of them, in my view, measure all of biological aging. And I think it remains to be determined how useful each of these tools are individually at measuring biological aging if your goal beyond just a funny thing that you can look at that gives you entertainment value. If your goal is really to use these as endpoints in clinical trials, for example, or to predict future health outcomes, which is what I think they might ultimately be useful for, we just don't know at this point which are going to be useful for doing that. How do we get that information, I think, is through these sorts of studies where we can actually measure various parameters that we think might reflect biological age and actually follow individuals over time and show that indeed these things with some level of precision at the individual level predict future health outcomes. And more importantly, we can move those things with interventions and they predict the effect of the interventions on future health outcomes. So I think dogs are an opportunity to do that in an accelerated way compared to what you can do in humans, because dogs age much more rapidly than people do. Absolutely, you can develop an epigenetic clock in dogs that works very similarly, apparently, to epigenetic clocks in people in terms of, giving an estimate of chronological age and maybe some predictive value for biological age, or at least future health outcomes. What we need to do now is show that we can actually validate those at the individual level and for interventions that they are actually predicting future health outcomes at the individual level. That hasn't been done yet. I also this is a little bit of a definitional thing, but I think it's useful for people to understand. You can use those things in clinical trials as endpoints, but they are exploratory endpoints. We are far away, I believe, from being able to use those things as surrogate endpoints that any regulatory body will accept as a surrogate for the things that really matter, which are mortality, future health outcomes, quality of life. I think we'll get there, but I think we're farther away from that than most people recognize or understand. The reason being, you got to prove that they actually work before a regulatory body is going to accept those things as a surrogate endpoint.
Thanks, Matt. I mean, really, really great question, and Matt, really brilliant answer. Important to note as biological age testing is becoming more and more common, for us to have a conversation around that. So thank you. We're going to move on to Corby Simpson. Corby, if you don't mind popping off mute and asking your question.
Sure. Yeah. Thanks, Simon. Can everybody hear me?
Yep.
So my question is less on the scientific side and more on the social side. And that is, we see a reluctance and pushback to the idea of tinkering with human aging and people that are like, I'll never have an implant and technology and things. But on the other hand, it's very socially acceptable to chip our dogs, and spend thousands or even tens of thousands of dollars to extend our pets' lives comfortably. So the question that I would have is, do you think that the acceptance of pet longevity will help normalize the idea of human longevity? Does one precede the other to move one forward?
Yeah, really good question. So absolutely, that was certainly something that I recognized early on in thinking about the whole concept of the Dog Aging Project, that it could potentially be an important communication tool for helping the average person recognize, number one, that there is a biology of aging, number two, that the biology of aging can be modified, and number three, that we should modify the biology of aging in a positive way if we can, to improve health span and potentially lifespan. And yes, I think you're right that for some people, it's an easier conceptual leap to think about the benefit of doing that in their companion animal than it is to make the conceptual leap to think about doing that in humans. And it may be a useful tool to help people go down that road and develop an understanding of all of those things. I also think there are other ways to have that communication with the general public that will be important as well. But I certainly think that the Dog Aging Project and other people who are working in companion animals now to improve health span and potentially lifespan, I think that can play an important role in helping the general public understand, accept, hopefully embrace the idea that we should be trying to modify aging in a positive way to keep people healthier longer. And
okay. Thank you.
Thanks, Matt. I'm going to quite selfishly pick some of these because I'm curious to know for my own dog. We've got a couple questions related to dog nutrition. I think it's really interesting, the study that you're doing on time restricted feeding, intermittent fasting with dogs, but we've got some questions on whether one meal a day means dogs are fed more or less. We've also got a question here about the type of food, wet food versus dry food, fancy food versus non-fancy food. So I wonder if you've controlled for that, or how you've managed like the nutritional process in relation to dog longevity. Yeah.
So again, very, very, very astute question. So the data that I showed you, and we've published one paper on the once a day feeding, in that analysis, we did not control for type of food. We intentionally did not control for type of food. And we don't have information from HLES, we don't have information on how much food that is precise enough to be able to really look at that. So we're going back now, and there are people developing more extensive surveys around the type of food and quality of food that we'll look at, but that wasn't in that original analysis. Obviously, both of those things could be important. So what I can tell you is obesity clearly has an impact on health outcomes in dogs. That's not necessarily from our work, but we see it in our data set, and other people have seen that in dogs as well. And my personal intuition is that's at least part of the explanation for that once a day feeding result that we saw, that dogs fed once a day are indeed less likely to be obese. We also have gone back and have started to look at, to the extent that HLES looks at the diet quality itself, look at whether or not there are any big differences between dogs that are fed, commercial kibble versus commercial wet food versus cooked home food or raw home diet. Started to try to look at that. I don't think so. My feeling, and again, don't quote me on. Maybe I shouldn't even say anything, but take it for what it's worth. My initial reaction is the effects there aren't as big as what we saw with the once a day feeding versus more than once a day feeding. So there may be effects there between, say, raw food versus kibble, but they certainly aren't as striking as the difference that we saw with the feeding frequency. So I think we just have to look a little bit more deeply to be able to address that. And again, the last point I'll make is it is important to remember that data that I showed you was cross-sectional. So it was all based on prior disease diagnoses that owners reported. So it may very well look different with dogs that are in the study now as we go forward. If they're being fed once a day for a year, 2 years, 3 years, will that correlate with different disease outcomes in the future? It might look a little bit different with that sort of data compared to the cross-sectional prior disease diagnoses data. So it's just important to appreciate the nature of the data itself.
Great, thank you so much for that. There's a question here from Gail. I'd like to paraphrase it a bit, and it's about the longevity of Salukis in particular in the US, and how there's a reduction of longevity of Salukis based in the US and Europe versus their traditional kind of stomping grounds, which are desert bred. So have you looked into, let's say, initial dog environment versus the US and Europe and how that's changed? I mean, I know that there's always this discussion of, what were human beings bred for? What is we had the caveman diet, we had this. And there's this idea that our initial environment is what we should be, aiming towards in our contemporary lifestyle. So have we kind of looked on how dog breed origination translates to alternative lifestyles?
Yeah, so I will say we haven't within the Dog Aging Project, and I don't think there's actually great data out there. So this question actually just came up recently because a reporter contacted the Dog Aging Project and wanted to know if dog lifespans are increasing or decreasing, right, in just a general sense. And the answer is, after lots of back and forth, we don't know. So I don't know that there really is a good answer out there, even as to whether or not in the United States today, I'm not talking about Salukis specifically, I'm just talking companion dogs in general. Have we seen a significant increase in life expectancy among companion dogs, or decrease or change at all over the last 10, 20, 30 years? I don't know that there's great data to really answer that question, but within the Dog Aging Project, we actually can't address the question you were asking, because at least for now, all of the dogs in the Dog Aging Project have to be in the United States. And that's a infrastructure thing that is a source of ongoing frustration to me, and we are going to fix it at some point. But at least for now, all the dogs in our cohort are dogs in the United States. So we are limited to asking questions more around, rural versus urban versus suburban, or pollution levels, but within the United States. So I can't really answer that question. I'll also say, this is just my personal sort of perspective. I'm not particularly convinced about the idea that we should try to match the environment that we evolved for. Number one, I'm not sure we really know the environment that we evolved for. We can speculate. Certainly it was different than it is today, but I'm not sure there's any reason to believe that that's going to be a optimal environment from a longevity perspective. So I don't find those arguments particularly convincing in the case of human longevity.
Fascinating.
In the case of Salukis, what just might be interesting for you is you've got a subset, you've got a small population that, and in this case, the data available on Saluki longevity, say in the Middle East, might in fact, historical data might in fact be available.
Yeah.
And then enable a compare contrast. And what I've just heard, granted, some of the things I've heard, for example, European, I watched a bunch of Salukis develop cancer, but so did their people, and they were downstream from Chernobyl. So
Right.
go.
Right.
But
Yeah.
But in terms of just looking for things that, if there are environmental triggers that you can identify, hypothesize, have impacted that group, and then expand the study to your larger populations, it might be a very useful tool for gaining some insight.
Yeah, I agree. And I'll just, again, make the pitch, right? There's data out there for a lot of different purebred breeds of dogs that you can get access to. And our data is open source, right? So I absolutely encourage people who are interested in this to do exactly the kinds of comparison that you're suggesting. Get access to our data, get access to the pedigree data from purebred dog breeds in whatever part of the world that you can get access to that data, and ask those kinds of questions. So yeah, I hope people will do those kinds of things. And that's part of the reason for making the data publicly available.
Yeah, and there's probably a whole separate topic of conversation to talk about community research, this growing trend, which is truly incredible. I know we're a bit above time, but there's just one last question. There's been a couple of questions related to rapamycin. I know it's something you're highly involved in. A lot of people are curious about. I'm going to call on Camilla to ask her question and see. Yeah, there we go. Camilla's here.
Hi. Thank you so much for your presentation. It was extremely interesting. I was wondering what is known about the effects of rapamycin on the dog brain, because I came across of some puzzling data in mice showing brain volume reductions on MRI following 4-week rapamycin treatment. So did anyone did similar studies in dogs? And if not, whether you would be open to collaboration with preclinical imaging group at Oxford?
Yeah, great question. So, I mean, I think the real answer is we don't know yet, because nobody has tested before we did rapamycin in dogs, other than some studies on osteosarcoma in dogs. Those are the only clinical trials that I'm aware of. So the answer is we don't know, but we are certainly, first of all, absolutely interested in collaboration. So feel free to shoot me an email. And we can talk about that. What we are doing, there's two things that we're doing actively. One is collecting cognitive assessments on the dogs in the clinical trial every 6 months, using that tool that I mentioned. And also having the owners, and these are sort of more pilot pilot kinds of things, but having the owners do some of these games that Evan McLean and others on the team have developed that we think can are reflective of different aspects of cognitive function within the clinical trial. And then there's a veterinarian scientist at Colorado State University named Stephanie McGrath, who studies dementia in dogs. And she has it's actually a 4-arm clinical trial with dogs that have canine cognitive dysfunction, very small. So it's kind of I don't know that unless the effects are huge that we're going to be able to draw any strong conclusions, but there's a placebo group, there's a cannabinoid group, a cannabinoid plus trazodone, and a rapamycin group within that clinical trial that's part of our brain health study. So, we are trying to at least start to collect some data on whether rapamycin can impact brain neuropathology, biochemical markers of dementia in dogs with canine cognitive dysfunction as part of that study with Steph. So it's super interesting area. And like you said, there's a whole big body of literature from mouse models of normal cognitive decline during aging, as well as mouse models of Alzheimer's and tauopathies, suggesting that rapamycin or other mTOR inhibitors may be able to have some beneficial effects in that context. So definitely very interested in looking deeply at that as much as we can.
Okay, fantastic. And what about sex differences? Do you see any differences in terms of response to rapamycin between sort of male?
We don't have enough data yet. So again, yeah, really interesting. In mice, it is the case that female mice at a given dose seem to be more responsive than male mice to rapamycin. I'm not aware of any data in humans suggesting a similar trend, and we just don't have enough data in dogs yet to be able to speculate on that. So we're aware of it and absolutely going to pay attention, but I can't answer that question right now.
Right. So my question was sort of motivated by what we see in the context of metformin treatment, in terms of using metformin following whole brain cranial irradiation in mice. So what we noticed that it looked very promising in terms of alleviating the radiation-induced brain injury in female mice, both as an acute treatment and treatment sort of long-term following the injury. However, in males, the acute treatment exacerbated brain radiation-induced injury. This is work that we already published. I can send you that. So we're sort of very curious about this kind of like a sex difference, and we're seeing this kind of like brain volume reduction in males, and then we have this rapamycin data in mice, and we are seeing these brain volume reductions, and we're trying to understand what it really means and whether it translates to other
again,
species.
we could spend a whole hour talking about sex differences in aging. If you haven't had somebody on to talk about that, there are really great people you could have on to talk about that. But you're absolutely right. The difference of some of these interventions that have been tested in mice, even in male and female mice, you often get very, very different outcomes. Rapamycin in some ways, at least for lifespan, is relatively unique among interventions that have been found reproducibly to increase lifespan in mice, in that it seems to work robustly in both males and females. But even there, at a given dose, females seem to be more responsive than males. And those mechanisms are, really, really not well understood at this point, but obviously super important for translational. We need to understand it, certainly, when we start thinking about translational applications of these interventions.
Right. Thank you so much.
Well, Matt, that's a great idea, actually. I might follow up on that and ask you your advice on a speaker for that topic. Sure. But I mean, everyone, please join me in giving a round of applause for Matt. This has been really, really fantastic, really fascinating. I know I'm going to be sharing the recording with lots and lots of dog owners and colleagues in this space. I'm just going to drop, I hope it's okay, Matt, I'll drop your LinkedIn in the chat. Sure. I know there was some possible collaboration, so you can reach out to Matt if there's something you want to discuss. I'll also drop in my LinkedIn if you're at all interested in the Oxford Society of Aging and Longevity, getting involved, joining the committee, contributing in any way. We are always open. As I said, this recording will be online. So if you want to circle back to it, it's always there. Otherwise, Matt, this has been so generous of you. I know that we've gone well above time. Thank you so, so much.
Absolutely. Thanks, everyone.
Take care.
Great talk, Simon.
So great talk, very interesting. I hope to be in touch with both of you guys later, you know,
Thank you. Appreciate it.
Absolutely.
yeah. Fascinating, yeah. Very interesting.
Well, looking forward to it, Gail.
Great, okay. All right, see you guys later.
Take
Bye.
care.
Matt, I'm so sorry about the host thing.
That's all right. This is a Zoom thing, but I don't know what it is. I don't know if it's just, a Mac thing, but every time I've had that happen before, and it's, you're trying to use the pointer, but I can't get rid of the window, right, that shows admit this person. And so it was a little bit confusing, but we got through it. It's all right.
It's just so shocking, there are these tech companies that have unlimited funds, and very obvious design flaws.
Yeah.
Like for their core, and yet they don't get fixed. But thanks for bearing with that. Sorry,
Yeah.
I know it's the multitask
problem.
sometimes.
It's
Well,
it's
listen,
good
we'll
for me.
follow-
It engages my mental exercise there.
Yeah, well, this is a longevity,
That's
intervention.
right.
But listen,
All right.
we'll follow up on email. I'll send you the recording once we have it again. Thank you so much. This
Cool.
is really generous of you.
Yeah, no problem. All right. Have a great day.
Take care. Ciao.
Bye.